So good morning, everybody. Welcome to the Humanist Community in Silicon Valley Sunday Forum. My name is Matt Courtney, and I'm the recorder and a member of the board of the Humanist Community. The Humanist Community is a chapter of the American Humanist Association. Humanism is a reality-based philosophy of life that affirms our ability and responsibility to lead ethical lives of personal fulfillment that aspire to the greater good. We value freedom, health, happiness, fairness, compassion, and using science and reason to acquire and apply knowledge. If these words describe your thinking, we invite you to become a member of the Humanist Community if you've not already done so. Membership forms are available on our website at humanists.org. That's humanist plural. If you are listening for the first time, welcome, and we invite you to continue listening to our weekly forums and other events. You can find all our events listed on the website humanist.org. Please aid us in continuing to present these forums by donating to the Humanist Community. You can find out how to donate to our organization on the website, again, humanist.org. Our speaker today is Ron Garret, and he will discuss how we know the biology of biology and evolution. This is, I think, number 7 in his series on the history of science. So, Ron, you should be unmuted and go ahead and take it away.
Okay, thanks. Let me just turn on the screen sharing thing here. Okay, can everybody see the slide? Okay, I can't.
Yes, we can.
Okay. Whoops, I gotta make a little adjustment here because Zoom is covering a part of my notes. Okay. So this is part 7 in the series of How We Know What We Know. There's the context of the previous talks and some of the planned future talks, just to put this one in perspective. The idea behind this series is to talk about the history of how scientific knowledge is acquired and the processes behind it. Scientific knowledge is all too often presented as received wisdom. In this area of COVID, trust the science has become a commonly heard refrain. Well, you shouldn't trust the science. You should question the science, just as you should question everything. But you should come to believe the science because if you question it, you will find that it stands up to very harsh scrutiny. And the reason that it stands up to very harsh scrutiny is that the body of scientific knowledge that we have today is the result of literally centuries of humans applying such scrutiny to various ideas and keeping only the ones that do withstand scrutiny, and in particular the ones that are consistent with experiments and thus have the best correspondence to reality. So this series of talks is more about the process than the product. And I have to start this one with 2 disclaimers. First, I am not a biologist. This is the second talk in the series about a topic in which I am not a domain expert, the other one being the previous one about geology and the age of the Earth. Everything in this presentation comes from my high school biology education and information that I was able to find on the internet. So take that into account as you assess the reliability of what I'm about to tell you. Some of it may be wrong. It is almost certainly incomplete, particularly since, disclaimer number 2, biology encompasses an enormous body of knowledge. One of the things I tried to find out while I was researching this talk is how many biology papers are published each year. And the answer turns out to be that no one knows. It's a number that's certainly in the tens of thousands, possibly hundreds of thousands. As far as I could tell, nobody keeps account. It is actually impossible for a single human to get a grip on all that is happening in this field. In fact, for a while, I considered splitting this into 2 talks, one on biology and one on evolution, but the 2 fields are just too intimately related to be teased apart that way. And so maintaining any kind of narrative coherence, so we'll just have to try to squeeze it all in. So with that said, let's get started. We humans have been studying biology since prehistoric times because we, of course, are ourselves biological entities, and we're naturally interested in understanding ourselves. But biology, as it turns out, is unbelievably complicated. We still don't have anywhere near a complete handle on it even today. So the situation back in pre-industrial times was pretty hopeless in terms of making progress towards real understanding. If you've ever seen the movie The Nightmare Before Christmas, which I highly recommend, by the way, you'll remember the scene where Jack Skellington, the leader of Halloween Town, is trying to make sense of this newly discovered Christmas thing. And he does all sorts of experiments, including dissecting this teddy bear, which of course does not end well for either Jack or the bear. But this is not so far off from how early humans explored biology. There was a lot of dissecting and a lot of random experimentation, which usually didn't end well for the subject of the experiment, and very little actual progress. Now, to be fair to our ancestors, I should say that archaeologists have found evidence of prehistoric brain surgery. And the really remarkable thing is that the subjects often appear to have survived the process because the bone shows evidence of growing back. So they were not total hacks, but nonetheless, the dominant biological theories up through the mid-17th century or so were all pretty much bogus, having to do with spirits or humors or miasma in the atmosphere and a whole bunch of other stuff that we could spend several hours on. But I'm going to skip all that towards the beginnings of actual progress towards our modern scientific understanding, which is arguably the invention of the microscope by this fellow, Antony van Leeuwenhoek, and I have no idea if I am pronouncing that properly. I'll make that a blanket disclaimer too. There are a lot of names here that I may well be pronouncing wrong. Around 1670 or so, there aren't good records of exactly when he made this, when he invented the microscope, because the importance of what he was doing was far from apparent at the time. Van Leeuwenhoek was not a scientist. There was no such profession at the time. He was a draper, which is to say someone who sold cloth, and he started grinding lenses to make it easier to count the threads and otherwise assess the quality of a piece of cloth. His microscope looked like this. The sample was held at the tip of the little pointy thing. I wish there was a way for me to have a pointer that you would see, but that's something we'll have to work on for next time. The lens is the little hole that's just above the tip of the point, and you use the thing by holding it up to your eye, kind of like a jeweler's loupe. I want to read you this quote from one of the sources I found, because it's hard to appreciate the magnitude of what Van Leeuwenhoek did otherwise. Leeuwenhoek spent a considerable amount of time perfecting the manufacture of lenses for his microscopes, and he was able to grind and polish biconvex lenses to an amazingly high quality. It is also suspected that Leeuwenhoek used blown glass lenses, and that these lenses were the ones responsible for the incredible magnifications of his simple microscopes. Leeuwenhoek produced these lenses by chipping away the excess glass from the thickened glass droplets that formed at the bottom of a blown glass bulb. These incredible lenses had a thickness of about 1 millimeter and a radius of curvature of 0.75 millimeters, so about the size of a BB. They had superior magnification and resolution when compared to the other microscopes of his time. The Utrecht Museum has one of Van Leeuwenhoek's microscopes in its collection. This incredible instrument has a magnification factor of about 275 times, with a resolution approaching 1 micron. And I've got a link down there in case you want to follow that and learn more about his microscope. They really are amazing instruments, considering the technology that he had to work with. In any event, in 1674, Van Leeuwenhoek became the first human to see a microbe. He didn't know what they were, but he saw that they moved, and so he guessed that they were little tiny animals, and he was essentially correct. So now we have to radically shift gears and mention this fellow, Carl Linnaeus, who lived in the 1700s, about 100 years later. He was the father of modern taxonomy. Taxonomy is the study of categorizing and labeling living things. If you've ever taken a biology class, you will probably recall the mantra of kingdom, phylum, class, order, family, genus, species. That is taxonomy, and it began with Linnaeus. He published this work Systema Naturae, in Latin, of course, because all scholarly work at the time was done in Latin. Over 10 editions, at the end of which he had cataloged 4,400 species of animals and 7,700 species of plants. And we still use his system of categorization today. Around the same time, this guy, Jean-Baptiste Lamarck, advanced the first theory of evolution. Now, you have to remember that Lamarck knew absolutely nothing about genes or DNA or heredity. He just observed that biological organisms reproduce and that the offspring are very similar to their parents, but not completely identical. So he hypothesized that over time, these incremental changes could accumulate to produce all of the features of living organisms, and that this happened because physical traits were passed on directly from parent to child. A classic example of this is the neck of a giraffe. On the Lamarckian hypothesis, giraffes stretched their necks out to be able to reach high up into the trees for leaves to eat. And so their children would have longer necks as a result of this stretching. We now know that this is wrong, of course, but it was not an unreasonable hypothesis given the data he had to work with. And he was kind of on the right track with his idea of incremental changes accumulating over time. But the big piece of the puzzle that Lamarck was missing was the idea of natural selection, but I'm getting ahead of myself. The next character in the story is this guy, Gregor Johann Mendel, who was born a year before Lamarck died, just to give you an idea of the pace at which progress was being made in these days. We're now into the 1800s. We're still very far from our modern understanding of biology. Mendel is famous for the heredity experiments that he did on pea plants and for discovering the existence of dominant and recessive genes. An interesting bit of trivia about Mendel, he almost certainly falsified his data because it was too good to be true. If you go back with a modern understanding of heredity and look at the results that Mendel got, they are an almost perfect match for his theory, and the odds of getting such a perfect match are very close to zero. So it's possible that he got lucky, but much more likely he fudged his numbers. At the sort of opposite extreme of Mendel, who gets more attention than he probably deserves, is this contemporary of his, Dr. John Snow, who's hardly ever mentioned at all outside of scientific history geek circles. Snow was a physician in London during a cholera outbreak in 1854, which killed about 500 people. That sounds like a big number, but to put this in perspective, the previous year, cholera killed about 10,000 people in and around London. Cholera outbreaks were a regular occurrence back in those days because no one knew what caused them or how to stop them. There were two competing theories of what caused disease back then. The dominant one was that diseases were caused by something called miasma, which was associated with bad smells in the air, which kind of makes sense because disease is often associated with bad smelling things. The other theory was the so-called germ theory, which said that disease was caused by these little microscopic critters that Antoine van Leeuwenhoek had discovered about 200 years earlier. Snow is famous, to the extent that he is famous, for plotting all of the incidents of cholera on this map. This is a copy of Snow's actual map. The little black bars are his notations about where the cholera outbreaks were. And you can see that they're clustered around the center of the map, a 2-block long street called Broad Street. So he hypothesized that the source of the cholera was not the air, but the water coming from a water pump that was on Broad Street, that the surrounding residents used as their water supply. He managed to convince the Board of Governors to take the handle off the pump so that it could no longer be used, and in so doing, he probably saved thousands, if not tens of thousands of lives, because, of course, as we now know, cholera is a waterborne disease. That's the result of raw sewage seeping into the water supply. But at the time, this was a very controversial idea. And even after Snow's intervention successfully stopped the 1854 outbreak, the germ theory remained controversial for many years after. This is, there's actually a memorial to John Snow at the corner. This is a reproduction of the pump that was there whose handle John Snow had removed. And if you look in the background, there's a pub called the John Snow. Anyway, the first person to successfully convince the scientific community that the germ theory was true was this guy, Louis Pasteur. Pasteurization is named after him. This is a photo of Pasteur in his lab. He's famous not just for inventing the process of heating food to kill bacteria that we're all familiar with today, but also for this ingenious series of experiments that demonstrates the existence of germs. It's worth diving into this in some detail because it really was a turning point in the history of our understanding of life, and also a turning point in scientific methodology. So at the time, it was widely believed that life arose spontaneously whenever you had the right environment, which was usually some kind of rotting food source. It was already known that if you took a sealed container of some food and heated it up, that it wouldn't spoil. But the advocates of the spontaneous generation theory argued that this was not because there were germs, but because sealing the container made it impossible for air to get in, and this lack of air is what prevented things from growing. So Pasteur invented this thing called a swan neck flask, which is just that. It's a flask with a long curved neck like a swan. The neck is open, so it lets the air in, but the curve prevents microbes from getting in. So he was able to do this experiment where he started with two identical flasks full of beef broth. He heated them both to kill anything that was already living there, and then he removed the neck of one of the flasks. And the broth in the flask whose neck had been removed spoiled, and the broth in the other one didn't, despite the fact that both were open to the air. And they also did this third experiment where he tilted the flask with the neck intact so that the broth was exposed to the air at the end of the swan neck, and that one spoiled too. And this is one of the first examples of a properly controlled biological experiment. The results are reproducible, and the difference in outcomes can clearly be assigned to a single variable. So we're now in the mid-1800s, and what is arguably the most momentous event in the history of biology, which is Charles Darwin and his publication of Origin of Species in 1859. So let's pause for a moment and put this in historical perspective. 1859 was the same year that Pasteur did his swan neck flask experiment. So Darwin actually didn't know about that result. Germs were still controversial. DNA hadn't been discovered yet. That wouldn't happen for another 10 years. And even then, it was just another chemical. Its role in heredity would not be discovered until 1952, almost 100 years later. So by the standards of what we know today, Darwin had next to nothing to work with. And yet he had one of the most beautiful and prescient and, as it turns out, correct ideas in the history of science. It really is on a par with relativity or Turing's theory of computation, and it's described right in the title, On the Origin of Species by Means of Natural Selection. The idea is that everything we see in biology can be explained in terms of a process of random mutation followed by selection for reproductive fitness. It's an idea that can be described in a minute, but to justify it, to show that it's actually true, takes a lot longer. Origin of Species is a thick book. It is chock full of data, including, of course, Darwin's famous finches, which is what originally inspired him. In case you don't know, in 1835, so 24 years before the publication of Origin of Species, Darwin was on an expedition to the Galapagos Islands, which are in the middle of the Pacific Ocean off the coast of Ecuador, and he noticed that there were different species of finches living on the various islands, and none of these species were known to live anywhere else in the world. So there were only two possibilities. Either all of these species had somehow gotten to the Galapagos independently of each other and then somehow died out wherever they originated from, or they all somehow originated on the Galapagos. And then he further observed that these finches were not evenly distributed. Certain species were found on certain islands and other species on other islands, and all of them seemed to be well adapted for the particular environment in which they lived. The most likely explanation, and indeed the correct explanation, as it turns out, is that these species all descended from a common ancestor which came to the Galapagos from somewhere and probably still exists wherever it came from. In fact, we now know that it is most likely this bird, called the blue-black grassquit. And the descendants of that original colonial bird adapted over time to the various islands. And the crucial insight, the reason that Origin of Species is such a fat book, is that all of the diversity of life can be explained this way, not just these finches. I'll give you just one example of how we know that Darwin's theory is correct. The ultimate test of any scientific theory is its ability to make verifiable predictions, and Darwin's theory makes many. Here's one that Darwin personally made. There is a species of orchid that grows on the island of Madagascar. It's called Angraecum sesquipedale. And it's distinctive because it has a flower with this really long part that dangles down, which is where the nectar is stored. Darwin predicted that there would be an insect that evolved to take advantage of that flower as a food source, and 150 years after he made that prediction, exactly such an insect was discovered. It turned out to be a moth called Xanthopan morgani, and it has this absurdly long tongue, just long enough to reach the bottom of that orchid, which is now called Darwin's orchid. But anyway, back to our story. I'm going to have to start leaving out a lot of stuff because there's just too much of it. But one of the highlights is undoubtedly the discovery of deoxyribonucleic acid, or DNA, in 1869 by Friedrich Miescher. Miescher, of course, had no idea what he discovered. He was just working on cataloging the different chemical compounds that he could isolate from cell cultures. The people who did figure out that DNA was the carrier of hereditary information were Alfred Hershey and Martha Chase. And they made this discovery in 1952, so less than 100 years ago, about 70 years ago. They did an experiment using radioactive isotopes of sulfur and phosphorus to track the migration of proteins and DNA during the reproductive cycle of something called a bacteriophage, which is a virus that infects bacteria. And by the way, this is the reason that this discovery took so long to make, is because the technology required to make it hadn't been developed until then. The radioactive isotopes, all the research on radioactive isotopes had been spurred on by the advent of World War II and the race to develop the atomic bomb. So you often find in the history of science this interplay between the development of technology and scientific discoveries. Anyway, so the experiments were based on radioactive sulfur and phosphorus, because proteins contain sulfur but no phosphorus, and DNA contains phosphorus but no sulfur. So Hershey and Chase took some radioactive sulfur and fed it to some bacteriophages, and some radioactive phosphorus and fed it to some other bacteriophages, and then let them reproduce, and the offspring of the phages with the radioactive phosphorus were radioactive, but the offspring of the radioactive sulfur-tagged phages were not, and that shows that phosphorus, and hence DNA, is transferred to the next generation, but sulfur, and hence protein, isn't. So now we knew that DNA was the carrier of genetic information, but we still didn't know how this information is encoded. That discovery is famously credited to Francis Crick and James Watson, who published the physical structure of DNA in 1953, just 1 year after the Hershey-Chase experiment showed that DNA was the carrier of genetic information. Now, notice that I didn't say that Watson and Crick actually discovered the structure of DNA. They get the credit, but in fact, a lot of the actual work was done by this woman, Rosalind Franklin, and her advisor, Maurice Wilkins. But it was 1953, and in 1953, women didn't get credit for scientific discoveries. So it's not until fairly recently that that Franklin and Wilkins' contribution to the process has been recognized. Anyway, they used a technique called X-ray crystallography, where they shoot X-rays at a sample in a machine like this one. This happens to be a modern one. But they shoot X-rays at a sample and look at the resulting pattern of X-rays coming back out of the sample and work backwards from that to figure out what the structure, the atomic structure of the sample is. This is what the pattern for DNA looks like. If you do some fairly hairy math, which I don't have time to get into, you can figure out that this is the pattern that a double helix makes when you shoot it with X-rays. This is Watson and Crick back in 53 with the model of DNA that they built out of metal. So in rehearsal, I was running out of time at this point. I guess I'm talking too fast. But I'm getting to the end. There is a lot more to say about this, and maybe I should have added more, but I guess we'll leave time for Q&A. So just a few more key people that I want to mention. This guy, Georges-Émile Palade, is the guy who discovered the ribosome, which is the piece of the cell that transcribes RNA into proteins. It looks like this. It was also done by X-ray crystallography. Richard Dawkins is famous, or perhaps infamous, for being a militant atheist nowadays, but what actually put him on the map as a scientist was his 1976 book, The Selfish Gene. This is the cover of the first edition, and also the author photo from the first edition. So this is what he looked like back when he was actually doing this work. Up to that point, there was a controversy in the theory of evolution of what exactly natural selection was selecting for. Was it individuals or groups or species or tribes or something else? And Dawkins showed that, in fact, evolution selects for the reproductive fitness of genes, which is an entire talk in and of itself. And great, our gardeners are here. Finally, I want to mention this guy, Kary Mullis, who invented the polymerase chain reaction, or PCR. And I mention him not just because PCR is a crucial tool that greatly accelerated the pace of discovery in biology in general and is today used in COVID tests, but also because he's a really interesting character. He won the Nobel Prize for discovering PCR, but he is not your stereotypical stodgy scientist. He was a surfer and kind of a hippie. And he also subscribed to some really fringe ideas. Most notably, he was an HIV denialist. I subscribed to the borderline fringe conspiracy theory that the HIV virus is not the cause of AIDS. And that's a whole other talk that I could give. Like I said at the beginning, biology is a huge topic, so but I've run out of prepared material, so I have to stop there and open up the questions.
All right. Thanks, Ron. Don't worry about stopping early. Like, apparently everybody does it. There's something about Zoom that makes it makes these talks go a lot faster than everybody expects.
Yeah, I think it's because when it's in person, people occasionally will raise their hand in the middle and there'll be a pause for questions, which will launch some impromptu discussion of something. and that adds time. So I need to revise my page count to time estimate conversion for Zoom.
Well, that's okay. We'll have more time for questions. So I already have a couple hands up. So first one is Chip. Go ahead and unmute yourself.
Okay. So right in that series with Watson and Crick, right before that was a discovery that I think was quite critical, that of the 4 bases of DNA, which can be measured, 2 of them track and 2 of them track. Some species have more. I think it's AT is a pair and GC is a pair. Some species can be up to 60% AT, 30 each, and 20 each of GC. But the discovery that they always track as 2 units implied the cross connection at the core. Does anyone know who that was?
I have no idea. I thought that was Watson and Crick also. I thought that discovery came along with the discovery of the chemical structure. But I don't know. Do we have any actual biologists with us? I guess not. It surprises me. I think you'd find some here in the Silicon Valley.
Yeah. Chip, did you have more comments?
Just that I had read the matching numbers in the DNA types had occurred before Watson and Crick.
Oh, that's certainly possible, because that's a discovery that you could make without knowing the chemical structure. You could just measure the relative concentrations of them. So yeah, it's quite possible that somebody figured that out earlier. But again, I have no idea.
And to have been born in an age with magazines like Science News, where I was getting these sorts of results, the fresh results in the field. The communication is so key to coming to an understanding of this stuff.
And the recency of it all is just staggering. You know, to think it was only 70 years ago, which is easily within living memory, we didn't know how DNA worked. And now sequencing DNA is a commodity. You can have your entire genome sequenced by 23andMe for $50.
Well, they deliver a scattershot sample.
Yeah, right.
They're not doing any. You actually don't get any PCR from 23andMe.
Right. I'm exaggerating a little bit, but still, you know, the fact that you can get any information about your genome at all for $50 is just that blows my mind.
All right. I got to move on to other people. So, Alex and Senna, a question?
Yeah. Pardon my ignorance here, but the last thing you mentioned, what is TCR? What does it do?
PCR is the polymerase chain reaction. And it's the process by which you take a little tiny sample of DNA and amplify it so that you can tell what's in it. That's the first step in sequencing DNA.
I see.
So it was a technological development more than a scientific development, but an absolutely crucial one. It was the development that it's kind of analogous to van Leeuwenhoek's microscope. It opened up this entire field of DNA sequencing from which vast quantities of biological knowledge are derived today, including the sequence of the COVID virus, which led to the development of the vaccine. And the way that the current batch of vaccines were made was by starting with the genetic sequence of the COVID virus and then engineering an RNA strand that match the spike protein on the COVID virus and then producing that and injecting it into you so that you develop antibodies that match the structure of that protein and block it, prevent the virus from being able to enter your cells.
Okay, thank you. You might want to say something about CRISPR too.
Yeah, I don't know much about CRISPR.
Okay.
I'll say what little I know just for people who have never heard of it. CRISPR is another recently developed technology that allows you to edit DNA. So it's basically a technology that you can use like a little tiny pair of scissors to cut and paste strands of DNA and rearrange them to produce the sequences that you want. But that's all I know about it.
Yeah, the frightening technology.
It can be. All of this is potentially very scary.
All right.
Especially if you like to watch a lot of the late night horror movies.
Yeah, we gotta move on here. Got a lot of people with their hand up. So Susan and Roy.
Yes, I want to mention to the idea of CRISPR. I read in a scientific magazine, American Scientist, that CRISPR was really part of biology as far back as the Neanderthal. It's a process that really started with the human genome. So anyway, and let's see, let's see, I'm thinking, Chris, okay, I have another question, but I'll let it go and I'll raise my hand again.
Well, if anybody wants to look it up, CRISPR is spelled CRISPR, no E. It's an acronym. I don't know what it stands for, but in this age of the internet, I don't want to do it because I want to focus on the Q&A. But any of you could just go Google it and read up on it and tell us what you found in real time if you want.
Oh, by the way, Chip, I just want to mention, maybe this is obvious, but when you have 4 base pairs and they pair off, you end up with binary logic. So when you see the deviations of genome, it's in the...
that was actually done chemically very early on. I threw the word PCR out, so I believe I should explain it to everyone. Consider 2 inputs. One is a soup of G bits, A bits, T bits, and C bits that you blend up, and it is the components of a DNA. The second input to the process is a long coiled up DNA pair. So it's 2 strands wrapped around each other. The chain reaction has 2 steps. The first step is you apply chemistry means to cause the DNA to unwind into a single-stranded DNA. And the second step is you pour a pint of these mixed up A bits, C bits, T bits, and G bits, and let them find their match on the uncoiled strands. And what you have done is you have doubled your strands. You took an initial left and right strand, you peeled them off from each other, and you duplicated them up into 2 coiled strands. So each time through this sequence, the chain reaction is a step of uncoil yourself, and here's a pint of goodies, and rebuild yourself, and uncoil all of those selves, and then they rebuild. So every time through, you double the amount. And so it's a beautiful scientific process to understand and how it does what it does.
All right. Chip, did you have a question on top of that? You had your hand up.
CRISPR was mentioned. One of the things you can do with manufactured DNA is you can build a probe that will you apply a drop of someone's blood or DNA or whatever, and the probe will detect, yes, I have a match for this many characters. And in biology, there are also proteins that cut DNA and proteins that paste DNA and proteins that repair DNA. And if you combine a probe with a cut and repair protein, you get CRISPR. So it has something to find and something to do when it finds it. And I agree, it's scary.
Yeah. Any comment from Ron?
Chip knows more about this than I do.
Yeah, on the comment that that CRISPR came from biology that's been around since Neanderthals. I don't know how long it's been around. I do know that it wasn't something we just made up. It was actually discovered, and I thought it was taken from like a virus or something. I'm not sure if that is true or not.
So I actually took a really quick look at the Wikipedia page. CRISPR, it turns out, refers to two different things. It is a kind of gene sequence. So it stands for clustered, regularly interspaced short palindromic repeats.
That's a completely different thing.
Well, it's not a completely different thing. It's the basis for the CRISPR technology. So there's the CRISPR gene sequences, and then you scroll down to the bottom, and you get to CRISPR gene editing, which has to do with these CRISPR sequences. Exactly how they're related, I haven't yet stumbled upon, but in any case,
the short tandem repeats are a DNA feature that DNA sometimes gets a chunk of this and then 18 more copies. And it turns out that once it goes over that and becomes 19 more copies, you get a disease. So it's very, very subtle how this stuff comes together. But oftentimes in DNA, you find these patterns of here's 40 of these. And CRISPR is good because it allows you to find one of them and cut it out. So that's how it grew from the study of CRISPRs. Their goal is to cure the disease by cutting out a bit of your DNA.
All right. I'm going to move on to Dana.
Oh, sorry, I forgot to change the name.
Jerry.
Yes. So I had one question. So you mentioned that we've been using the same taxonomy for a couple hundred years or so. I don't know I don't know much biology or much about this DNA, but it seems to me as though DNA analysis would let you understand things better to the point that maybe it would change your understanding and change the taxonomy.
It's not the exact taxonomy. It's the same taxonomic system that Linnaeus used, but of course our understanding of how things should be grouped improves over time. So the actual categorization of things changes over time, but the system, the hierarchical system of categorization, and the fact that all these species are given Latin names and the basic naming convention and all of that, the system goes back to Linnaeus.
Okay, but knowing and being able to study the DNA has enabled changing the categories.
Oh yes, of course.
Okay.
Right. Herman.
Okay, thank you. It's interesting that you mentioned the work of von Leeuwenhoek developing the first microscope. You might ask why that microscope, well, microscopes of today are not used to visibly observe DNA. And the answer is that the resolution, the smallest thing you can see with any source of light, has to do with the wavelength of the light. And visible light has a wavelength for blue or ultraviolet down to slightly below 1 micron. And DNA, to observe DNA, you need 100 or 1,000 times shorter wavelength to get the resolution that you need to look at the atoms that make up the DNA. And this was done with X-rays, as you showed with the X-ray photograph by Rosalind Franklin, whose photograph was analyzed by Watkins, Crick? Watson and Crick. And it's one of the unfortunate results in science where the person who should have gotten the credit for taking that photograph, Rosalind Franklin, did not get the credit. Watson and Crick actually stole her data. They took that picture from her and analyzed it and came up with the conclusion that it was a double helix and got the Nobel Prize for it, whereas she didn't. But the technique she used of X-ray crystallography is still in use today, and in fact it has been revolutionized by the development of X-ray sources that are 1 million times more intense than the conventional sources that Rosalind Franklin and other scientists used, which are very little different from dental and medical X-ray tubes. We have X-ray sources now using relativistic electrons bent in magnetic fields to go in a circle, which shed X-rays that are 1 million times more intense than conventional X-ray tubes, because the X-rays are very collimated and well-directed and very intense because they originate by a different mechanism than those in an X-ray tube. The problem with an X-ray tube, such as a dental or medical tube, is that the X-rays are not directed. They come out in all directions, which is why when you take a dental X-ray, they put a lead apron on you because the room is filled with X-rays, and you don't want any to get on other parts of your body because X-rays are not good. So a tiny, tiny fraction of the X-rays produced by a dental X-ray machine go to making the picture of the cavity in your tooth. The rest of them give no information, and in fact could be damaging. You note that the technician who takes the X-ray ducks behind a lead glass screen, and you're covered with a lead blanket. So the field of X-ray crystallography, which is so important now in understanding viruses like Ebola, like HIV, like COVID, are studied intensely at laboratories like ours at Stanford, which makes use of high-energy electrons circulating in magnetic fields to generate these very intense X-rays. Rosalind Franklin may have taken days or weeks to get the pattern that you showed, whereas it can be done in a second or so with modern X-ray tubes. So it's interesting to note that during the concerns about COVID, laboratories such as ours at SLAC, Stanford Linear Accelerator Center, have been shut down because we don't want people getting together, and yet the use of their X-rays is so important in understanding COVID that they have been operating those facilities which are used by scientists to understand COVID in the 4 such laboratories in the United States at Stanford, Berkeley, Argonne, and Brookhaven, Long Island. All of them have been shut down or for the general purpose scientific studies, except for ones related to COVID using X-ray crystallography. So X-rays are an extremely important means of understanding the atomic and molecular nature of materials, such as viruses.
By the way, I'd like to point out Greg Alexander has been posting some very worthwhile comments in the chat. And one thing in particular that I'm a little bit chagrined about, he pointed out that everything that I've talked about has been the history of biological science in Europe and Western culture. And there's quite a bit of stuff that's happened in Eastern cultures that I completely ignored. And I'm sorry about that. I feel chagrined.
Something to add to your presentation next time, right?
Yes, absolutely. And thank you, Greg, for pointing that out.
All right, we have Fran and Jim. It actually just looks like Jim. You got to unmute yourself, Jim.
Okay.
There you go.
I just wanted to add to Herman's comments that X-ray crystallography has advanced to the point where they can determine the structure of a great number of enzymes and other very important proteins. And if you look at a modern biochemistry or molecular biology book, you see this proliferation of all these protein structures and very advanced structures like the ribosome and such that have all been derived from X-ray crystallography and some very involved algorithms, I suppose. I'd also like to mention that CRISPR is actually was invented by bacteria in probably hundreds of millions of years ago. The fact that viral phages, as was pointed out, invade bacteria and kill them. But when they inject their DNA or RNA into the bacteria, the bacterium in defense would cut out a portion of that DNA and attach it to its own chromosomes. So the next time a virus would invade the cell, it would recognize that DNA and had devised this protein protease that would cut out the DNA from the ribonuclease that would destroy the DNA of the invading virus. So it served as their protective mechanism. And this was discovered in the laboratories at Berkeley and developed into the CRISPR process.
Jim, I'm sorry to interrupt. I need to duck out for just 1 minute. I need to print out a check and give it to our gardener. I will be back. This literally will just take a minute. I'll be right back.
All right. Thanks, Jim, for that update. I knew it had something to do with viruses. I didn't realize it was actually the bacteria that came up with it and as a protection mechanism. Looks like Ron will be away for a little bit. I don't see anybody else with their hand up, so I'm thinking we should move on to general discussion. And when Ron gets back, we can ask more direct questions. I'm going to go ahead and allow everybody to unmute themselves, but looks like Dana and Jerry have a hand up.
Still muted.
Yeah. Roy pointed out that that the DNA is binary because each step of the way you either have an AT pair or a CG pair. But I was wondering that if you took an AT pair and swapped where they are on the double helix, you move each one to the other side, so you got AT gets replaced by TA. Is it unaffected? Is it exactly the same? Different?
That's one of the cases. I mean, when you get a de novo mutation in a cell, a base pair is replaced, either swapped or changed to the other kind and swapped or swapped. So there's three possible places to move for every case.
Right. So if it just was swapped, so AT became TA, does it act differently?
Oh, yes, it will completely remake the protein.
Okay.
That's what's happened in the variant from South Africa. One pair was mutated, changed one protein to another. One amino acid to another, and apparently completely changed the configuration of the binding site on the virus, which makes it much more contagious and perhaps more lethal.
That is one of the features of the success of a pandemic. The more people that have the pandemic or have the virus, is the more laboratories that evolution can explore to change the virus. Its very success produces the mutations that make it more successful.
Just to be clear on from the math side, I think DNA is technically like quaternary. I don't know exactly what the actual proper name is, but it's base 4, not base 2. But because 4 is a power of 2, it's very easy to represent it in base 2. I see.
So
Ron is back.
Does anybody know the mechanism by what goes wrong when a mutation occurs? I mean, some copy mechanism must be failing in some way. Does anybody know where that failure is?
There are a number of known processes, the proteins that part of it is that there are repair proteins as well as there's a copy protein that runs down a DNA strip, pulls it apart, sets another out, and that's how you get RNA reading. And in each of these cases, there are known steps that can go wrong, which result in de novo alteration of the original DNA.
Although it's important to point out that most of the variation that happens from generation to generation happens because of the mixing and matching of genes that happens during sexual reproduction.
Yes, meiosis is much more susceptible to mutation than mitosis.
But even if the process is perfect, sexual reproduction produces random variations because every generation you get a combination of two different genomes, one from the mother and one from the father. And this is the reason that sexual reproduction is so prevalent. It's because it makes the process of mutation much more efficient.
Or it encodes the process of mutation in the very act of survival.
Or the act of reproduction. But yes, and also because it in some sense narrows the search space to genetic sequences that have already proven themselves to be useful in the process of survival. So if you just start randomly flipping bits, you are as likely, more likely to end up with a mutation that will decrease your reproductive fitness, or to the extent of potentially even killing the organism.
I got a statement about that. I read in the Scientific American that COVID virus is the only virus that has error correcting logic. Just like they have a computer built in. If the error correcting logic is very powerful in COVID, why does it have mutations at all, the variants?
Many viruses have it. If it's there in COVID, it's also there in SARS.
Yeah, that's error correction is not uncommon. The ability to mutate is not uncommon. In fact, it's ubiquitous. All life forms do both.
Apparently,
that's a contradiction. That we have error correction and then mutations easy. That seems to be counter logic.
Let me explain.
We're talking about a lot of viruses.
Now, let me explain the method by which sexual reproduction forces evolution. If you take, we have 23 chromosomes, but for every chromosome in mitosis, you produce 2 baby chromosomes in each daughter cell. And the process in meiosis is directed to allow evolution in this way. To run through meiosis, which only occurs in sexual reproduction, it is the method to produce the gametes that must later come together to produce a totempotent cell to grow into the next generation. So gametes have half your chromosomes, but the way they are built is that you take the number 2 from your mother, or the number 8 from your mother. You take one of the chromosomes from your mother and one of the chromosomes from your father. Now he had 2, but only one went into his meiosis. So the gamete that starts you is half. When you produce your own gametes, you take your 2 chromosomes, one from mother, one from father, you divide both of them in 2 and produce 4 possible gametes. But that's when the fun only begins. Granted, right there you have the capability to produce a varying evolutionary thing where each chromosome has its own survival characteristics. And you produce 4 because you have 4 inputs. However, there is also something called crossover, where you lay down a chromosome and you twist them like pretzels, and you decide that from here to here, I'm going to take Papa and put it in my first gamete, and Mama goes in my second gamete. And then further along, it's the other way around. And they swap back and forth on the length of the chromosome during crossover in meiosis. So meiosis is the process of producing gametes, and it only matters to sexual reproduction. Every other division, every other cell in your body grew through mitosis, the simpler way of copying. So you have to understand meiosis and crossover to see how what I call the Cambrian explosion, people say about that, you've heard that term? I call that the invention of sex.
Well, I'd just like to point out that that life in itself is an incredibly, astronomically improbable occurrence. It must, for the first cell to have formed, there must have been an extremely a great number of highly improbable reactions. First, to form the components of a cell. The simple bases, the simple carbohydrates, the lipids that go into the membrane, all of these things are magical chemistry. It's completely improbable that a cell should have ever formed. And the improbability of it is exaggerated by or emphasized by the fact that nobody's been able to do one under carefully controlled conditions in a laboratory where they must have been trying dozens of different combinations of temperature, chemical components. And
so this is the question of abiogenesis, and it's not as improbable as you think. So the first thing to realize is that the first replicator was almost certainly not a cell.
Oh, certainly.
It was almost certainly
like an RNA.
It was almost certainly something like a strand of RNA. And there's actually been some interesting recent developments on this that I happen to know about, because I do a lot of debating of young Earth creationists. So this is something I actually do keep track of. There is an artificial model of a minimal self-replicating system. And I forget the details offhand. I have to go back and refresh my memory. But there's a certain number of things that have to come together in order to make this hypothetical artificial model happen. And you can do the math on the probability of this happening by accident, and it's pretty unlikely. But the thing you have to remember is that in the early Earth where this happened, there was no competition for this early replicator. It was just an entire planet full of organic molecules bouncing around. And so the odds of it happening at any given place at any given time is extremely low. But the odds of it happening somewhere on the planet over a period of, say, 1 million years is pretty close to 1. And 1 million years is not a very long time. It's less than 1/10 of the age of the Earth. So there's the odds are actually very plausible, even though we don't understand the details of exactly how it happened, and we haven't been able to reproduce the process because we don't have a planet-sized laboratory in 1 million years to wait for it to happen again. But there is a very plausible mechanism by which it could have happened.
I grant that there is a plausible mechanism because there's billions and billions and billions of experiments going on on the planet in these millions of years, for sure. But
it must be acknowledged that it took almost 1 billion years after life was clearly here for multicellularity and the invention of sexual reproduction to occur. So it takes 1 billion years for a lot of bacteria to work out how to do sex.
I kind of wonder about the evolution of DNA. You know, it's a great chemical. It's got the advantage of being very stable. It's got the disadvantage that it doesn't do anything, but it can be converted to RNA. So RNA somehow figured out how to generate DNA to preserve its status, its culture, or whatever.
So this is actually also fairly well understood, and it's another thing that the creationists don't seem to grasp and constantly get wrong. Reproductive fitness is not an absolute measure. It's always measured with respect to an environment. And in the very early Earth, that was a very particular environment where RNA could survive on its own because it didn't have any competition. Relative to an environment and relative to your competitors in that environment. So in the early Earth, where you essentially have no competitors, RNA by itself can thrive. But as soon as you start getting this reproductive process going, you start to get competition. And once you have competition, being able to partner with a molecule like DNA that's more stable gives you now an RNA molecule that's partnered with a DNA molecule a competitive advantage over your competitor molecules that don't have this ability. And so all of these are very easily understandable as incremental steps of incremental improvement in reproductive fitness relative to your competitors in your environment at the time that change occurred.
I think DNA is kind of like the invention of writing.
But where did the DNA come from?
That's the question.
That's a big
that's
step.
almost the beginning
That's
of
a good step from RNA, and
obviously intelligent aliens planted it here.
Of course.
I would say that also there may not have been a competitor for RNA, but RNA could be very easily hydrolyzed. That's the big problem with forming polymers. They're not essentially stable in many environments, and RNA is hydrolyzable. So it would take particular conditions to produce these replicators. And I do believe that RNA was the original replicator and the basis for life, because we find in the cells now that there are enormous numbers of variant types of RNA that perform all manner of roles that we have very limited knowledge, although they do moderate the activation of genes and such, and they also can modify the proteins that are produced from a particular gene by the way they're cut and spliced and changed in length, and which produces a different frame for amino acid incorporation, which would mean a different protein. So it's a really complicated system, and you really have to kind of look into it before you understand how enormously complex a cell is. Having worked in a laboratory and trying to make a simple compound in a flask with adding components and such, what you usually wind up with is a fairly small yield of the product you want and a lot of tar in the bottom of the flask. The cell manages to produce all these enormous numbers of products essential to life, and without without a lot of waste and byproducts. And it's an elaborate system of feedbacks and augmentations of certain enzymes while others are suppressed. And it's just an incredible machine. And for it to come into existence, in my opinion, is very improbable. And I think our chances of finding life on other planets and such is going to be very surprising to me.
Well, the question in my mind is can you have complex reproducing life founded on something other than DNA? And I would think there is. DNA is a nice molecule by molecule code. I suppose, in a sense, if you made, what was it Turing had the idea of a self-replicating machine that would read a tape, that would tell it how to build copies of itself, and then run a copy of the tape and feed it in.
I think you could do it with RNA.
RNA is not stable.
Well, it's not as stable as...
It combines with everything.
Yeah, it gets worn down.
Which, by the way, that's the reason that the vaccines have to be kept at ultra cold temperatures.
Yeah.
Well, they're also protected too in lipid membranes or lipid micelles, I presume. But I would think that life could evolve where RNA is protected and could be the reproductive material. It works in viruses. So.
Well, one easy way to protect it is to switch one of the molecules in there and form a double helix and surround it with sugars and whatever.
Well, an interesting possibility is to someday, possibly within the lifetimes of some of the people here, we will be able to build a self-replicating mechanical system.
Yeah.
And so like a 3D printer that's able to print another 3D printer. And that is a very scary prospect, because now you've essentially created a new life form that's non-biological.
Yeah, instead of DNA, you'll have a binary memory code, a charged or uncharged location.
Yes.
The game was called Core Wars. You had to build same and battle it out on a unprotected memory.
But Core Wars was a simulation, and I'm talking about the possibility of having actual physical self-replicating mechanical systems.
There is a self-replicating, I mean, life was created. Someone said, everyone talks about how all life comes from life. I'm going to, in a laboratory, put together the necessary chemicals to make a living thing. And they did. And in a protected and fed environment, this cell that they put together, which is more of a clump, can replicate and survive. It's a very simple creature and would not last long in the real world. But
I doubt very much that that's true, because if it were true, it would be much bigger news, and everyone would.
It's only a few 100,000 chemicals he puts in. It's nothing like a real cell.
Doesn't matter how simple or complicated it is. The artificial creation of a self-replicating system would be one of the biggest breakthroughs in the history of science. It would be.
I thought I read
news.
about it 2 or 3 years ago.
Oh, you may have.
Let me dig for a minute here. I'll see if I can find it.
Yeah, I seem to remember somebody creating like an artificial cell as well.
I can imagine somebody claiming that. I would be very skeptical that the claim.
It
stood up to
seems to
scrutiny.
me, I'd read something a long time ago about certain chemical combinations. If you have the right soup, you can put one molecule of something in it, and it will rearrange all the other molecules to copy itself.
So what has been demonstrated is the synthesis of amino acids from basic chemicals that were plausibly present in the early Earth. Mm-hmm. And yes, the process that you just described, which is essentially a sort of a crystallization process.
Yeah.
But the creation of an actual self-replicating organism from scratch, that has not, like I said, I'm not an expert. It's possible that this happened. I haven't heard about it, but I'll give you extremely long odds against.
But isn't this crystallization a molecule copying itself?
No, because you have to then, the thing that you create has to then be able to go on and make yet another copy of itself, right? It has to be this exponential process. It's not enough to do something like you get in, say, a supercooled liquid where you introduce a nucleation site and the thing suddenly freezes and the crystal structure of the frozen material spreads throughout the material. That's not life, because you're not copying information.
Isn't the structure itself information?
No, because the structure of that sort of process is built into the material that you're freezing or that you're crystallizing.
They replaced all the DNA in E. coli.
Okay, I remember that one.
Yeah, that's possible. But that's very different than creating a self-replicating system starting from scratch.
But they did have to use the environment of the cell, I think, to carry it out.
Right.
And another one they did is they expanded the genetic code in some work they've done. So that's a new form of life, but that wasn't from scratch. Let me keep digging. I think I saw this many years ago.
XNA. I think that was on one of what was that? Fox Mulder and Scully and Mulder thing.
Lab-made microbe, a strain of DNA of bacteria normally found in soil, created a living organism that has a fully synthetic and radically altered DNA code.
Yeah, but that's still very different.
I think they may be using envelopes from life. That would be cheating, in my view.
That's not
using
really
envelopes from life.
That's not just an envelope, it's all the mechanisms within that cell, too.
Yeah, it's kind of like
Read and redesigned. Okay, this is about the E. coli work. Let me keep going.
It's kind of like starting with a 3D printer that's already capable of printing another 3D printer and reprogramming it to produce a different kind of self-replicating 3D printer. It's not the same as producing a self-replicating 3D printer from
Or building an engine for a car body and calling that creating life in the car.
Cars are not really a good analogy because they're not self-replicating.
I imagine also in the DNA they injected this original DNA. They probably also had to copy sequences from recognized DNAs in order to create proteins and such that would be essential for a new type of organism.
So I'm going to have to go here fairly shortly. If anybody has any more questions they want to direct to me, particularly before I bug out.
Nobody else has a question on the 3D printer, self-replicating 3D printer. I'm pretty sure we can replicate everything, but just like a few pieces of the 3D printer. It's already set up like that. Maybe the little the engine that moves everything.
We're nowhere close to being able to make a truly self-replicating 3D printer. You might be able to make a 3D printer that can print, say, a chassis, but you would have to actually make the business end, the print head. You'd have to be able to print that.
That's true.
And then you'd have to be able to, and then you have to be able to assemble it all.
Yeah.
Right?
No, it definitely doesn't assemble it all, but I know like there are people who have it printing like everything else on the 3D printer, including like the bars and stuff, but they're all made out of plastic, so they're really not durable. So hardly anybody does it except as an experiment to show that.
Well, you can print something that looks like a 3D printer. But the tricky part is you need the print head, and the print head contains things like heating coils and
that are smaller than the print pixel.
Yeah, they're made out of different materials and have much tighter tolerances, and it's nowhere near being a solved problem.
True.
Well, Ron, thank you very much for that very enlightening talk. And really appreciate the depth of your knowledge in all these many different fields.
Thanks for having me. It's always a pleasure.
Yes. A whole list of names to go check on Google and find out more about them.
Ned, I'd like to have an introduction to the guy that's looking over your right shoulder.
That to me, that's everybody should know who that is. Hopefully. No,
he passed recently.
Yeah, it's Amazing Randy.
Oh, really?
It's not done yet. His hand still needs work, but most of the rest of it's done. I know it's kind of a skewed angle as well, but yeah. If we ever get together in person again, I'll bring it to our meetings.